This is a microtiter plate, and in these small wells we create artificial microbial ecosystems under very controlled conditions to study ecological processes. During my studies at the university, I found it extremely interesting to work with this tiny microorganisms, which are so relevant for many processes. And it was fascinating for me to realize that in a few grams of soil, we have thousands of species. We know, for instance, that bacteria, which are so essential for many ecological processes, are embedded in rather complex networks. And within these networks, we find competitors, we find partners, but there are also quite a lot of different predators like protists, bacterial predators, and importantly also viruses. We discovered, for instance, in microtiter plates, in these mini ecosystems, that species which seem to be functionally redundant under certain conditions turn out to be much more important if we change the conditions or under stress, which means we have to take into account each single species, actually. We would like, of course, to understand how these networks function and how predator-prey interactions affect essential functions in an ecosystem and also adaptation to global changes. And for this, we have to take the findings of these mini ecosystems and compare them with reality. That's why we are also working in field experiments, such as the GCF in Bad Lauchstädt, where we can follow with sequencing tools, for instance, the adaptation of networks under realistic climate scenarios. Society could benefit from this research if we would be able to apply this knowledge on networks, on predator-prey interactions for a sustainable agriculture, for instance, or for alternative ways to control pathogenic microorganisms. Yeah, let's take, for instance, microbes. They have really a bad reputation despite their really deep importance for humanity, for the globe. And I believe there's quite an interest in the society to learn more how we could use these microbes and the microbiomes for the benefit of all of us. And I think there we have to take this opportunity and the chance to transmit and transfer this microbiological knowledge into society, into schools, for instance.